Okay, I'm going to make an attempt at recording this right after school gets out. We're going to see if I get interrupted. Um, this is actually going to be the last lecture of this unit, covering proteins, the last of our macromolecules. And they're all essential, I think. Proteins are the most interesting. Let's jump right in. So, of all the macromolecules we've talked about so far, proteins really kind of have the biggest range of what they can look like and what they can actually do. Generally, if something is happening in your body, if you're doing anything, it's probably done by a protein facilitated by other proteins. They're involved in defense, like our immune system, and transport, getting stuff around and communicating and moving and support, everything. We can even break them down for energy. So they really do have a huge range of actual functions. They are made up of, however, just basically 5 elements. The usual suspects of carbon, hydrogen, oxygen, and nitrogen. And this is where sulfur comes into play, that we'll talk about, maybe not in this unit. The monomer for a protein is an amino acid that you can see up here. If you string some amino acids together, there's announcements. There's a new teacher meeting at 3 p.m. in Ms. Pugh's room. So monomer is amino acids. You string them together and it becomes a peptide. When the peptide folds and gets its shape is when it actually becomes a protein. We're going to talk a lot about these. So the building block being amino acids. There are 20 main different types. There are actually more than 20 amino acids, but 20 is what mainly is what we use and what most things use. And they're characterized by 3 things. They have an amino group. You can see on the left here. They have a carboxyl group. This is where the acid comes from, so amino acid. And then they also have an R group, or this variable side chain. And that can kind of be everything, or anything, excuse me. And that is what we actually use to determine which amino acid is which and to categorize them, is what is on this R group. And based on that, we put them into 3 categories. So they can be polar R groups, they can be non-polar R groups, or they can be ionic. They have an actual charge to them. You'll see a plus or a minus in there. And that gives us an idea of kind of what kind of molecules they're going to interact with, what kind of chemistry those particular amino acids will have. So you can see the non-polar at the top, polar in the middle, charged at the bottom. You don't have to memorize these. What you do need to know is that we distinguish them by these side chains and the chemistry of those side chains. If you swap a non-polar for a non-polar, sometimes nothing happens. If you swap a non-polar amino acid in a protein for a polar amino acid, that can cause some big changes. So it can be helpful and good to know, like, if I change between these groups, big changes are going to happen. If I make a change within these groups, probably not much is going to happen chemically. So we have an amino acid. We need to link them together. We do so through yet another dehydration synthesis. It forms a special bond called a peptide bond. It's just a bond between amino acids. And yeah, basically we chain a bunch together and you get a polypeptide if you have multiple chains joined together. And we do determine there's like an endedness to them, kind of like with DNA. So if it ends in an amino group, it's the N terminus. If it ends in the carboxyl group, it's the C terminus. Not super important, but could be interesting. But it does give them a directionality. So you can actually, like, there's a direction to read the amino acid. It's read from the amino terminus to the carboxyl terminus, from left to right, essentially. And we always add amino acids to the carboxyl terminus. You can't add amino acids to the other end. It can only grow in one direction, just like DNA. I like how my subtitles capitalized one direction because it thought I was talking about the band. R.I.P. Liam. So, polypeptides, again, if you just join enough amino acids together, you get a polypeptide chain. You can represent it like this. More often, you'll see it like this bottom one where they basically just tell you what the amino acid is. You get a 3-letter code, and so on. Again, don't have to memorize anything with the codes. What we really care about proteins, though, is their structure. Remember, shape determines function, and so the specific 3D shapes or conformations that these proteins will have will basically give them their function. If you change that structure, you're going to change their function. Usually, you destroy the function. So if they're not folded correctly, they're usually non-functional. There are 4 levels of structure. We're going to talk about them each, from primary, secondary, tertiary, up to quaternary. So primary level, the easiest, I think, it is just the sequence of amino acids. So what order, writing them out. With just 20 amino acids, you can build hundreds of thousands of different polypeptides. So there's a lot of variety here. If you change this structure, you're going to change all the ones above it as well. Theoretically, if you just know the order of amino acids, you can predict the final structure of a protein, but that is actually a huge unsolved problem in biology currently. Secondary structure is where we actually start getting some 3D shapes. You'll hear these as alpha helices, coils, or beta pleated sheets, just like folded paper essentially. And the important thing to know with secondary level is that it is hydrogen bonding in the backbone. So it has nothing to do with the side chains. It is hydrogen bonds formed between the backbones, so the carboxyl and amino groups of nearby amino acids. So when we lay chains next to each other, the hydrogen bonds will form, and it will form them into either these sheets or helice structures. You kind of just need to know that those two are related to secondary structure, and again, that it is from the backbone of the peptide, not the side chains. What is from the side chains are tertiary. So tertiary level, this is probably where like the most interesting stuff happens, the most unknown stuff. It's the final folding, the final 3D shape of the protein. And this is determined by those side chains, by those R groups. And this is where we start to care about, do we have hydrophobic R groups that are going to want to hang out next to each other? Are we going to be forming any hydrogen bonds between different amino acids that are very far apart? Is there going to be any ionic or covalent bonding going on between side chains? This is where sulfur comes in. It makes that disulfide bridge that's very strong and adds a lot of stability, which is why we need sulfur. Otherwise, our proteins become unstable, less functional, which is not a good thing. But key things here, 3D folding, this is where the side chains matter. So this is where you get all of those chemical interactions happening. You're never going to be like asked to predict, but you should understand the idea that if I swap this hydrophobic amino acid to a polar amino acid, I'm gonna cause a different shape. But if I swapped this hydrophobic one for another hydrophobic one, I may be okay. I may not actually even have a different protein shape. It may keep its function. So usually our hydrophobic amino acids tend to orient to the inside and all kind of group together in a glob on the inside, and your hydrophilic ones will be on the outside and orient towards water. So if you swap those one for the other, again, you're going to get change in shape, change in folding, because that thing that wants to be on the inside is now on the outside. And then finally, we get to quaternary structure. This one doesn't even apply to every protein. It's only when you have 2 or more protein chains that fold together to form one big protein. We then call the individual peptide chains subunits of that protein. Big example is hemoglobin. It's made up of 4 subunits, and each of them individually don't carry oxygen. All of them together do. So it's kind of just when you get multiple different chains that are working together, folding together, that may not have been chemically bonded. You could produce 4 of these chains in separate ribosomes, and then they all fold together. So not everything will have this, only certain ones, and it is characterized by multiple polypeptide chains. Everything before this was a single polypeptide chain. So overall, we have our primary structure that is just the sequence of amino acids. This leads to some secondary structure of the helices or the pleated sheets based on hydrogen bonds of the backbone. And then the tertiary structure is that twisting, bending to get all of our side chains happy, hydrophobic with hydrophobic, hydrophilic with hydrophilic. And then quaternary is multiple chains, 2 or more polypeptide chains. We can alter our protein structure, as I've kind of been hinting at. It can be a mutation, so you can change the DNA. You can just have errors in copying it. But if we change the primary structure, we're definitely changing the primary, obviously, but it will likely lead to changes in the secondary, tertiary, and quaternary structure as well. Whereas if I change something about the tertiary structure, it may not affect down. And again, it is especially likely if you switch them with something to an amino acid with different properties. So again, switching a polar amino acid with a non-polar, and so on. So for example, again, hemoglobin carries our oxygen, and it is 146 amino acids in a single subunit. And if you change just one of them from glutamic acid, a charged group, yay, interrupted again. Oh no, they're practicing prayer. I'm going to try and finish this quick. And just talk over it. So if we make this change, you get sickle cell anemia, the blood cells get a completely different shape, and you get a bunch of health issues that can actually even lead to death and always lead to a very reduced life expectancy. We'll talk a lot about sickle cell anemia this year, so stay tuned. It's important to know that mutations don't always change the function of a protein. Sometimes, again, if you switch them with amino acids with similar chemical properties, nothing is going to change. And then denaturation, actually, we're going to get into in a future unit. They've changed how bio works. Same with these types of proteins. Don't need to know them yet, but they'll come back up later. So that is it. That is our lecture for the unit. Apologies for the announcement interruption. Sorry, it's just the end. I didn't want to re-record my whole video. That's all, folks. Yeah, bye.